And first of all, I would like to welcome all colleagues here to this meeting, and especially to our Director General, that even he's having busy schedule, he is here. So please join me in welcoming Dr. William Dar, Director General. And then, of course, Dr. CLL Gowda, other members of the management groups and research committees. So we are very much pleased to see you here. You can see that house is full. And just now I was seeing that how many colleagues have joined virtually by KSIConnect. So I think that some colleagues from ESA, WCA and other places they are there. So I would like to welcome those colleagues also who have connected us through KSIConnect. So welcome all of you. You all have seen the seminar announcement that we are having very special seminar today. And this is from Dr. Farid Waliyar. So all of you are aware about Dr. Farid Waliyar, but in case some colleagues they are new, and let me give a brief background about Farid. He is a principal scientist of plant pathology at ICRISAT since 1986. Prior to joining ICRISAT, he was a CGIAR staff in France, and he served ICRISAT in various capacity. And the last one was a director for West and Central Africa, and also AIC Managing Director, acting Global Team Leader for Biotechnology, Groundnut Team Leader, ICRISAT, Country Rep in Mali, etc. So at many places. In terms of his background, he holds a PhD in plant pathology from University of Paris, Mary flavus and other soil fungi on groundnut and their evolution during the cropping season in Senegal. So basically, what I want to say, so he's having a very long-term experience in this area of aflatoxin and in this particular area. As I told, he is having expertise in plant pathology, development of integrated disease management, identification of resistant varieties, and of course, while doing this work, he wrote several papers, more than and in different journals, books. I think more than 250 publications are there to his credit. Supervised several PhD, master's degree student, and he has received several awards, and I would not include or mention everyone, but would like to mention the most significant, and this was from the Mali Ministry of Agriculture in recognition of his contribution to development of agriculture in Mali. And as you know that Farid will be completing his term or services from ICRISAT on September 30th. So we thought that he is having experience for more than 30 years in this institute and the different areas. So let's have a seminar where he can update, or where he can give us his wisdom, his guidance to the new generation, that what we need to do, how we need to do. And we are very much pleased to have Farid with us. So please join me in welcoming Farid for this important seminar. Farid, please.
Thank you very much, Rajeev. As you can see, I am formally dressed, so I will be formal for a few minutes. And I would like to first welcome the Director General of ICRISAT, management member, the incoming Director General, I see he's here, and a member of the board. So many colleagues from Asia and also in Africa. So I am very pleased to see you here. I think most of you knows me and put me in a box, and that box is the aflatoxin box. I have done a lot more work than just aflatoxin, but again, I have a lot of experience. And the Director General of ICRISAT, Dr. William Dar, a few months ago, said, Farid, you are here for the next 6 months, and what do you think we should do in the 6 months? Well, I had an idea of what I'm going to do. It was trying to summarize some of the long-term data and write papers. But then he said, no, you should also think of putting some ideas on what ICRISAT need to undertake in future. Then when I proposed this to Rajeev, he said, well, this is good, but I really would like you to also give a seminar on aflatoxin research that you have done, so that everybody is updated. But then again, we had further changes because this would have been too long for one afternoon session. So we divided the presentation into two parts. The first part was discussed on last Wednesday with a number of colleagues in trying to see what are the future kind of work ICRISAT can do in this the area of aflatoxin. And as we have been developing within discussion, collaboration with all of the group involved in aflatoxin research here as well as in Africa. So that was discussed on Wednesday, and we came up with a number of suggestions. They came up so that I would then next maybe 15 days, I try to put those, take in account those suggestions of the colleagues and submit to the management of ICRISAT a version of the project or a concept note rather that ICRISAT could undertake next 5 to 10 years to some donors and maybe get the necessary funding. So here I'm really going to focus on some of the work done on the aflatoxin research. And although I'm going to make this presentation, it is not me at all. It is not me alone. It is a group of people who have been working together, and I cannot dissociate myself from them, particularly from the breeders, because this is where we have put the emphasis on for last number of years, well, 20 years or more. So I would be giving you a little bit of information on the aflatoxin problem, effects, and regulations, so that you can see where we are standing. And then I will give you an example of a study that we did in Mali. So that's some quite recent work, so that they can see where we are in terms of aflatoxin at the country level. And that at each country level there is really a lot of work to be undertaken. Then I would be talking about the aflatoxin research at ICRISAT for last 30 years and more. Then bring some of the achieved results, because honestly it is very difficult to summarize all these years of research. And then I would be talking a little bit about the diagnostic tools, ELISA, that many of you know, but some interest has been other day expressed again during the board meeting. And then see some of the future prospects. Aflatoxin is a very serious food safety problems that you will see across this presentation. In the past, many countries, particularly sub-Saharan countries, were exporting 50-60% of their product to Europe, from West Africa particularly, and today, well, a few years ago it was almost nil, and because this is because of the aflatoxin problem, and I will show you the way the level of toxins is allowable level of toxins are today in different countries. You know, AFB1 is the most toxigenic toxin, then followed by G1 and B1 and G2, G1 and B2 and G2. Very often we have tendency of forgetting the others, because 90-95% of the problem is from the B1. It's very dangerous, but still the total aflatoxin matters, and you will see in the most of the countries' regulation that is well reflected. And because AFB1 is also the most carcinogenic toxin, and a lot of work has been done. I'm not going to give you details, but the problem is of has two sides, you know, the health side and trade side. The health side, the trade side, I just was mentioning the restriction on trades, and this I'll show you a few minutes later why. Because the limits the allowable limit is very, very low now today. And of course the quality reduction. The quality reduction is something, you know, when the peanut or corn or whatever crop is really affected by aflatoxin, there is no way someone can eat it because the taste is changed, the color is changed, you cannot use it. So the quality reduction is one. But most complicated part is the health issues, because for very, very long time, not much information available. And then finally, when we got now we have a much better understanding of the problem. It is related also to malnutrition that causes all these problem. And very important, it is synergetic with the interaction with B and C, hepatitis B and C. In hepatitis B and C, for example, in Africa, is very, very common. It's very common, and before it was difficult to say, to make a difference, but today the methodologies are there to help understanding this and make the effect of aflatoxin on the liver itself much easier now. And then of course the cancer liver is the most common cancer, but there are many other cancers due to aflatoxins. Just to show you this, because this table, you know, this is the maximum allowable limits that many countries let you to import or export the quantity of aflatoxin. 10, well, it goes from 2 to 30. The European Union is for total aflatoxin, but B1 is maximum of 2 and then total 4. So this is extremely low. And I'll show you some data, and you will see how far we are from that reality. And some of the other, but you know, it varies from 2 to 30. In India today is still 30 ppb per kilogram. The other issue is really the complexity of the problem. It's extremely variable. That cultivars, the farming practices, and many weather conditions, whether it's drought or too much water, etc., every single factor will have an impact on the quantity of the toxin to be produced. In toxin itself, you know, I mean, the Aspergillus flavus, we know you have from 0 to 20, 30,000 ppb can be produced by different isolate of A. flavus. So there is big variability within this, and we are dealing with mixed isolates in the field conditions. So it is very much difficult to. So in among the you know, all these cultural practices, the harvest time, method of harvest, you know, if you harvest with what they call it in Africa, the daba is damaging the pods, and while removing, and this damage will of course increase the level of the toxin, and of course storage and other condition, packaging condition will increase the toxin limit. But the problem is many of the SAD countries, we have no monitoring system. There is not really well-established standards, and lack of awareness, and also lack of human capacity in infrastructure is also a part. Before I really give you some data or some results, I wanted to share this with you because this is something that we are, the focus of our work is, as you know, is groundnut. In many of West African countries, this is the value chain kind of situation, where you have the farmers from the farmers' field, farmers' granaries, and then it goes to traders one side, and some wholesalers also can buy from the granaries of the farmers. A part is going for processing, other for marketing, and then finally it goes to consumers. Why I'm showing this, we had a very nice experience with one of the IFRI, Gate Foundation funded project to IFRI. We were part of it, and we were able to identify in each of these critical points, we were able to identify what is wrong and what we can do. How much is the problem in each of these places, and you will see later. I cannot give, because this would require one full seminar if I give the detail of everything, but I will give you some examples. This is in Mali, 3 regions, and you see the number, the percentage of samples that can have different level of aflatoxin. So we have qualified this as a European Union, and then China, etc., USA, India, etc. So if you go by this, you'll see that, okay, in Kai you have 41% safe, but 59% is not safe. So like that, in Kai is only 36% of these products are safe, and in Kolokani 34. So like that, and you can see there are cases we have more than 500 ppb. So 500 ppb is a huge amount. Is not a joke. So this can kill people, and this is what happened in Kenya a few years ago. This kind of product, which have got thousands of ppb of aflatoxin. So the problem is serious, and then the problem will continue to worsen. And this is a case of farmers' granary in Mali. This is the level we have found at harvest time, but this is the level that 3 months later. It continuously growing because, as if you can see, this is in Mali, and the Mali Kai region is the driest. So the aflatoxin level is quite low, and it doesn't grow too fast compared to Kita, which is the most humid area in Mali, and then followed by Kolokani. So weather condition, that's one of the parameter, or one of the things that will condition this contamination or increase in aflatoxin in the granaries. So coming to what ICRISAT has been doing is that before the 90s, the focus was mainly breeding, and that time a number of resistant line was obtained, resistant in terms of germplasm. Some breeding material has been developed, and As you know, the breeding it takes about 8 to 10 years to bring before we bring a stable line. So in the 90s, the breeding lines they had anything between 20 to 50 ppb. But later, in the 90s to 2000, we continued the efforts on resistance breeding, and the level was less than 20 ppb. And this is because also of the job not only done by ICRISAT, but also by some of the US universities that have been bringing level of resistance to some better level. And then finally here, in the 2000, we changed our approach. We proposed a strategy on integrated management that was approved by the board in, I think, 2006. And we not only continued the breeding line, and we have, and I will show you some data, we have less than 10 ppb on those breeding lines. We have better information system in terms of value chain in its improvement. Kiran has been pushing a lot in terms of transgenic research, and also now recently transcriptomics research has been in place. So this is the evolution of our research in ICRISAT, and I'm going to give some of this example now. So this is what has been approved by the board. The approach is that we have a combination of host resistance, biological control, cultural practices, harvesting and post-harvesting technology that include drying and other technologies. Now, this is one side of the coin. The other one is then you have technologies that are no cost, low cost, and high cost. Because high cost technology is not for the poor farmers. And we, ICRISAT, we are really focusing on poor farming system, poor farmers, but there are farmers that they could take up those kind of technology, but we are in between these two and trying to, at the regional level, trying to implement those things and do the demonstration. If you just right now ask me where you have made real progress, I can say that we have made a lot of progress in all these areas, and I will show you, but we have been lumping here because transfer of technology is costly, and I will talk about this a little bit later. So in terms of host pollen resistance, we have been screening germplasm and including some of the minico, and I'm presenting some data on that area. We have been testing the advanced breeding lines in Asia and Africa, and of course incorporation of this resistant genes into locally adapted cultivars. Now, those are some of the breeding lines that we have been testing in Africa, but also the same set was tested here in Anandapur, in India, and I think we have sent the same set to Malawi. I don't know if Patrick is online, but I'm sure Patrick can update. But this is a set that has over time given extremely good results, and you can see in the very good control condition where you make sure that there is a lot of A. flavus available in the field. This is what is happening. But luckily in the farmers' field, they have been doing well, but we need to push this in terms of adoption and by most of the other most of the farmers in this region. So I was just mentioning the Minico, thanks to Hari Upadhyaya for his approach on identifying this Minico collection. We in 2008 already we had 184 breeding lines. Germplasm line for Minico, and we tested them in Mali for 2 years. After 2 years, we found that 36 of those lines had really good level of resistance. So we further test them in a more controlled environment. And we found that only 7 of them, this 7, have got really high level of resistance. Now, you can see our 6 years, this line has a little bit of 3.3 or 3.2, and this one has about 1 ppb. So this is excellent. And I hope our farmers, our breeders now can use this. It's a challenge for them, how quickly they can transfer these genes into locally adapted varieties. The other approach was a transgenic approach that Kiran has published a paper on how to make the transformation possible. And based on that, we used, he used the cartilage gene, and we developed a number of events that we have been testing. And already from there, we found that a number of them had less than 10 ppb aflatoxin. That is good, because this is on some of the GL24 variety, which is a susceptible one. And but still, we need to reuse this, and that was demonstrate the concept itself. The concept is working, but if you transform maybe a resistant variety, this level may be even close to 0. So a lot of hope in that sense, that this technology. And then Mrs. Kalyani, a student of Kiran and I, did further develop some events, and they are now well known as resistant. They have some level of resistance, as Kiran was saying. 80% reduction in most of those events. The other approach, the first one was antifungal approach, so that you stop the fungal growth. Here is to stop the production of aflatoxin. LOX13 is University of Wisconsin, Nancy's group had developed this approach, this LOX, and we had a collaboration with them, and Mrs. Saumani, one of our students, is she there? No? She's there. She has just finished her PhD, and I think she's going to submit it next week or something like that. So she has got some very interesting result on that work. But other work like defensins or phorisms are underway, and I think probably Kiran has to give you more information on this as they needed, but I put it here because I think there is one of futuristic kind of approach for the future. This would be one of the solutions that ICRISAT may have to take care. And this kind of approaches that in terms of agronomic practices that has been on the ground for some years. So we have been testing them for in a farmer's field condition for many years. Today we know that farmer manure and crop residue that is a kind of salt supplementers, and then gypsum or lime or whatever kind of sources of calcium you may find in the markets in West Africa, you can apply, and then that will increase the mechanical resistance of the pod. So we have been working in the combination of some of those treatment, and then we found after several years we have the farm-emulated lime is probably the most efficient, and we have been using this in some of the farmers' field, and for last several years in Mali. They were in Mali, Niger, Senegal. They are on demonstration, but really they need to be taken up on a larger scale to transfer. And again, you need money. Use of biological agents. I think we have been working for several years on Pseudomonas, actinomycetes, and trichoderma. We have really good potential biocontrol agents. They are efficient. And as you can see here, I mean, Dr. Rupila was working here to develop kind of compost with different trichoderma, bacteria, and different species. And we found that he found that it was, for him, it was just to accelerate the process of composting. And then he and I discussed, and we tried to see, is there any of this bacteria or that could also be antifungal? And then from that collection, we found a number of them having antifungal activities. And among them, we identify CDB35, which was good, and of course Trichoderma viride, which is well known. And we applied this here in combination with some of the other technologies like gypsum and in here at the campus. And very interestingly, all the 3 years we had really good level of reduction with trichoderma, but also with gypsum and bacteria combination. They had given really interesting result, and hopefully someone will pick up this. In among those actinomycetes, we find this, streptomyces, we found this CDA19, which was extremely efficient and suppressing the mycelia of Aspergillus flavus under the lab condition, but also in field. And Mrs. Harini, she did her PhD now. She has not yet published the paper, but there are some very interesting results on that. So we have a lot of good information available, and hopefully we can. But post-harvest, there is another issue that aflatoxin has been posing problem because. I'm asking all this question, and I have the response, and they are a big part of the solution. Even if you have a resistant variety that has been developed today, you take it to farmers and you put it in a bad storage with a lot of insects in it, it will be contaminated. So I think there is a lot of effort to be done in terms of post-harvest technologies. Just an example. This is an example in India. When they harvest in Anandapur, So you see, it is a rainy day. This will be full of aflatoxin. And then from there, after 4 5 days, they'll take it here. And we have tested inside of here. The humidity is very high inside of this heaps. And the aflatoxin contamination will grow. So one of the solutions we find, and we have been working with the farmers here and some of the NGOs in the south to go for direct threshing of the and then in West Africa, this is the way. And again, the same problem. The day it rains or the termites are too much in the soil, there will be a lot more aflatoxin. And that's why we have been proposing this to farmers. It's really not costly. It's just a question of spending a few minutes in taking care of those things, or making a small bench on the ground. So just to show that it's drying in drying matters. You can see these are we have been testing one susceptible, one resistant variety, and you can see you can reduce just by a very simple technology. You can reduce the problem by quite 84% in this case, or yeah, and then from the non allowable you become in the allowable level. You know, so there are a number of technologies that is possible that we can bring to farmers. So I'm just putting this because I think the three approaches, you know, host resistance, transgenic, or integrated management, there are a number of challenges. And I think, as I just mentioned, Rajeev, you are listening, I hope. You see, today we have this high level of resistance to some of the line. It has to be used. And I think you cannot use it if you don't have good markets. So I think we have to move quickly in that area so that development of genomic tools will be possible with those lines. But again, I am putting the seed system. Dr. Gowda will remember for how many years we have been working with the seed, but still we have not solution. So this does not mean we have to stop it, but I think if we don't have an appropriate seed system, we are not going to go far, because I mean, those improved varieties have to reach. In the case of groundnut, is a very difficult one because the multiplication rate is too small, and there is not much interest from the private sector. So there are a lot of problems that, but still, ICRISAT needs to work on those issues. In terms of transgenics, I think you all know the problem of regularity issues, lack of capacities, you know, in many places, a lack of private sector investment. So that in groundnut, I'm talking not about the other crop. So it is a difficult one. In terms of integrated management, there is no miracle. If you have no money, there is no way that you can go far. So I hope, because the research costs money, but also transfer of technology should cost money, and it costs money. So we need to invest in terms of transfer of technology. We have to put money. If we have not made such a huge impact that we wanted to make, is because we have not having the opportunities. But I think in West Africa now, from Ajita, with these three proposal that USAID is approving, I think there is a very likelihood of a big chance that we have some impact after 2-3 years. So I think that we should keep in mind and try to push our donor to give money. Awareness and policy support, I think it's evident, everyone knows, and we need this policy support, and hopefully ICRISAT will invest more on the policy side. I'm just putting this here because you all know that we have developed this ELISA test, which is simple, cost-effective, sensitive, rapid, and versatile. It has been helping a lot of countries. It has been helping Malawi. Moses is the one who has put a lot of effort and use of this technology to help farmers in Malawi. And they were able to export. But we can do more. We have more opportunities to do. And also, we have made more progress in terms of that. Anita, who is now a scientist of ICRISAT in Malawi, was my student here. And she did work with NIMS, Nizam Institute of Medical Sciences, to develop this in-house antibody for detecting aflatoxin from the blood. And that has a big, huge impact, could have in many areas. Not only this can be applied to other clinical trials in the medical institutions, but also in animal. And in fact, maybe Michael is here, and I think we have opportunities to work with ILRI and see how best we can together put some proposal together and come up in this paper. And I recommend you all to read. It's an interesting paper. It's been just published by Anita. I think I mentioned at the beginning of my introduction that we have been developing this technology, this project together, and we have been discussing with the colleagues. So we have now a proposal that I will be submitting soon to the Director General. I think, in my opinion, one way we have to focus on upscaling. Because we have a lot of results, plenty, and there is a big chance that we can make impact in both in Asia and Africa, but of course developing new technologies using modern tools. And that's why I think this project that we have developed is focusing on that in a lot of good innovative ideas are in that proposal. The other thing I would like to put here is that I have 24 years of data from West Africa, and then I explained that to Dr. Dar, and immediately he sees a lot of potential for that, and he suggested me a number of things to do. So we will try to do that and try to analyze the data on long-term analysis of our results. And but last 6 months already, 6 papers, peer-reviewed journal articles are in different stage of publication. I would like to, as I said, I had a lot of colleagues with whom I worked, so I hope we will continue this together, and I really thank all of them. To my students, I think they deserve, because they have had a important role in achieving some of the results we have. And of course to my parents and my wife and children. I just want to give you a personal note before I close. The aflatoxin research had a lot of highs and downs. There was a time ICRISAT decided to close even the activities on aflatoxin. for many reasons. But Dr. Dar in 2000, when he joined, and we had a discussion about the aflatoxin. So he asked me, so what do you have? So I explained to him what we have. And it is because of his really interventions that the research on aflatoxin picked up again. And Dr. Dar, thank you very much. A lot of press releases and a lot of communication, because awareness is a key, and he has been a key person to boost again the aflatoxin research. Thank you very much. Thank you.
Thank you very much, Farid. This was really very comprehensive and very informative presentation. So I think in case, he gave very good vision, and now we have some document with us, and I think we would like to follow up on that in coming months. But at the same time, if someone is having some clarification, something, we can request as well. Kiran Sharma is the first person.
Thanks, Farid. I think it was a very enlightening. I just wanted to know here, which is a more important or difficult part here? Is it the pre-harvest or the post-harvest? And how much does pre-harvest contribute when compared to the post-harvest? So that might give us some ideas, where to focus.
I think this is very difficult to say. I'll tell you why. Because if I'm analyzing the data with Upadhyaya on some of the germplasm that we have. 1 year you have 40 ppb mean of the trial, and another 1,800. So it's very difficult to say where the problem is more important. And we have cases in Mali, if you have, when you have say 200 ppb at harvest, you may have 220 after three months, but you may have as well 2,000. In some cases in Sikasso region, my work with my friends Mamouri Diarra showed very clearly some of the granaries is they are giving a lot because they are situated in a region where the insects are important and the humidity is high. That's why we didn't have much time because there is ICRISAT in collaboration with ICRISAT, we have developed a new granary where we use the smoke of cooking smoke that to dry the granaries every day. So, and we have found very interesting research. So already humidity in some of the region play a major role.
Okay, very good. Maybe now next question from Dr. Nigel Kirby, from our governing board member.
Farid, congratulations. It was an excellent talk. I think personally that it is very important work, and I'm speaking individually now, not as a member of the governing board. And the reason I say that is we cannot be corporately and socially responsible unless our food is safe to eat. So that's the standpoint I come from. If I may now, I'll ask you a more scientific and a very basic scientific question. I was wondering if you could tell us, or if there are any thoughts about what competitive advantage does the Aspergillus actually get from producing a toxin like this?
This is a very interesting question, because I asked this to some of our colleagues in IITA, because they are pushing this toxigenic strain to combat aflatoxin. The thing is that A. flavus million years ago was a non-toxigenic fungus. Over millions of years, it has become toxigenic. Reasons, difficult to say. But there is this tendency, and now today we have ICRISAT is keeping about 100, I think 100 isolates. So we have about 100 isolate here. Some of them are giving 25,000 ppb aflatoxin producing. So there is clear net difference. Now if you use this for biological control, my doubt is that they may become toxigenic anyway over time. So that way.
Okay, Dr. Hari Sharma.
Aflatoxins is also a problem in sorghum, and about 20 years back we did experiments to control the headbugs. We can reduce the aflatoxin by half. So I think in case of groundnut as well, if we have a good control of the mites, spiders, the wireworms, and then the storage elsewhere and all of that stuff, we can reduce the problem. So we could best and our crop post-harvest because of the
Oh yeah, no, absolutely. We have published some paper with Dr. Vincent Hume, who did study the relationship between the termite population in the field and aflatoxin contamination. Any damage, whatever is insect or mechanical or other, will increase the contamination.
Upender, do we have any question on the, no? Okay, good. Dr. Gowda?
Farid,
Yeah.
again, very useful information, research. I was just wondering that, in the presentation, you did not cover the aflatoxin as a control measure. Don't smile or laugh. No, the reason I'm asking is, that is being said as one of the effective ways of managing. Only 2-3 days back, I got a message telling that one of the countries has approved use of aflatoxin. So how good or not good it is?
I had a position. That's why I try to avoid responding. But you asked the question, I will give my response. This is a technology for rich people. Because it is expensive technology. The history of is called aflatoxin now. Is the same technology than Aflagord. Aflagord is in India for last 20 years. You know that? It's there, but farmers are not buying it. So one. The second thing is, you see, in the U.S., the way it was they started was the technology was developed in cotton, then brought into groundnuts, and then maize. And maize apparently is working well, but even there, where we are in terms of adoption of this technology in the United States, where you have richer farmers. So I think that should...
Yeah, related question. When I was in Nairobi and I had a discussion with Moses on our partnership with a number of centers there and with existing funding for existing activities, we touched about aflatoxin. Moses, can you elucidate what happened on the ground?
I wish I could come and explain in your office, but yeah, in Africa, aflatoxin is mainly being pushed by IITA, but as Farida said, there are two issues there. They are working more on maize because it's a crop that is not exported. It's a regional crop. So we have been a bit cautious about using it on groundnut since it's mainly exported. And for some of the countries, we really have no guarantees to say that it is offering the prevention that is claimed to offer. In terms of proposals, yes, we have a proposal right now in Malawi that AID USAID is funding IITA again to develop a product. Our frustration is that we have had a project in Zambia that is funded by USAID 4.5 years. We have half a year to go. We started the work, IITA started with Aflatoxin. They still have no product. Now they have developed now another proposal in Malawi and saying they have succeeded in Zambia. But in Zambia we know they have no product. So it is a moving target. As Farid says, really there is no product to show other than a lot of work moving from country to country. And
when you say no product, what does that mean? Because Aflatoxin is the product to promote.
Yeah, but Aflatoxin is the product that they have in the US. But they are trying to develop for each country a product of Aflatoxin. So for each country it is taking about 3 to 4 years for them to isolate the fungi that are specific to the country to develop the Aflatoxin. So they are not using the Aflatoxin product that they have from the US. It has to be specific for every country.
Okay. We have 3 more hands, so we will have quick question and quick answer, and after that we will close it. So I think, so one Dilip, then Manish, then Shobha. So yeah, one, you can have any order here. Dilip or Shobha, I think. Okay.
Thanks for a very good presentation. I have 2 questions. One is, do you know if Aspergillus flavus infest any other legume roots? Does it associate with any other legume roots? And then the other one is that with germplasm breeding, you have reduced the contamination with the known genetic basis to the extent possible. Would you predict 100% resistance?
Okay, Aspergillus flavus is a semi-saprophyte or saprophyte even. So it goes to any crop which is in a bad condition, drying, rotted, and so on. It can go. It can produce it can go in rice, it go in sorghum, millet, or any other crops. So it's a common fungal that is there. I don't know if we really have to go for complete resistance, because the danger is adaptation of the fungal to the new environment. So, or become more virulent, or become more problematic for other crops also. So I would not go for immunity now.
Okay, good. So DG has allowed that we can ask as many questions as we can. So we can increase the number now from 3 to 30 maybe. Okay, so maybe Dilip, Manish, Hari Upadhyay, then Ramjita. So let's do like that one. Yeah.
Thank you, Hari. Thank you, Farid. Such an informative and educative presentation, at least for me an education, because this is a new area. Yeah, one slide is very interesting and attracted my attention. That is technology transfer required funding. At the same time, another slide you have mentioned no cost and the low cost technologies. I'm more curious and interested to learn what are the major challenges, at the same time what are the major opportunities to improve the adoption rates in the farmers' fields. And at the same time, if we need to take up a technology transfer project, how do you prioritize the areas in the entire value chain? That would be very helpful, Farid, if you can throw some light on that one.
Yeah, thank you, Dilip. I think the major, the low cost, no cost is to farmers. The transfer of technology will cost to us, but no cost is to farmers. The challenge, you can divide the challenges into different categories, and I think one of the issue is to transfer the technology is to have tested first of all those technologies in the right environment. I tell you, the table you saw of those varieties that are doing extremely well, those varieties have been developed here in India. They have been tested here. but they have been also tested in Africa. And there, the result is extremely important. So that's the first thing is that we really have to have a good testing system. And then I think we have to be in a position to convince the governments that this is important. They have to be committed to solving the problem of aflatoxin. And this is a challenge because this government today is like the GMOs. Today someone likes and allow you to bring GMOs. Tomorrow someone come anti-GMO and will not. So aflatoxin being a problem that is not visible, many people are not convinced. In fact, even for the farmers, the technology we have been proposing to the farmers very often, we have not been telling them that this is for aflatoxin control, but this is just to give them better yield. That's easier to transfer a technology for. So, those kind of challenges are there, and the opportunities, of course, farmers, as soon as he gets something more in his plate, he will go for it. But the technology that just solve the problem of aflatoxin will not go.
Thank you, Farid. It's not a question really, and a marvelous presentation. What I wanted to say over here that these are really first time that we have a continuum of research on aflatoxin for last 15 years. Otherwise, when I came here in 1990, it was a dormant research. Continued for 3-4 years, and that's why when you see those 91 series line, 91, 278, 283, 284, they are the product of almost first 2 years only research from that. And then it was closed because of financial constraint. And then from 2000 onward, for 14-15 years, we have absolutely no problem. We have fantastic germplasm lines, we have excellent breeding materials, and these 7 lines which Farid did indicate, we are using them already in the breeding program. Not only in the Mali, but even when I see the papers from China or from US, whether it is 92305 or some other lines, which are of ICRISAT origin, are being used in US and other countries. And another point I want to make is that here, right now, the focus of the program, at least aflatoxin breeding, is not only aflatoxin resistance, but shortening the duration so that end of season droughts it avoids, as well as incorporating drought tolerance. So these 3 components ought to come together. Unfortunately, there is no premium either on release of the variety, whether it is having low aflatoxin contamination or not, and except in Australia, where some premium is there if your aflatoxin load is low in the produce. Other countries...
No, it's other way around. If you have aflatoxin, you pay penalties.
Exactly. So if your load is low, aflatoxin is low,
Yeah.
you get premium, or at least in Australia. Other places, whether Anantapur, it's loaded with aflatoxin or less, there is no premium for the farmers. And release also, there is no consideration for this trade.
Thank you. Okay, Manish.
Many thanks for giving such an informative presentation. I would like to know, because this is a very fast evolving pathogen, so have you come across there is some frequency, most frequent resistance breakdown?
No. I started working in Senegal in 1978 or something like that, I forgot. The first lines I was working there, it was 5547, one of the very famous varieties. That is still, if you have seen in the presentation, is still a resistant check. Okay, that its level is between 5 to 10 ppb, even that time, and it remains. The same has been tested in different places. There is no report of 55-437 or J11 to become susceptible in any place, no. We may have problem of isolate of the fungus that is more or less toxin producing. So sometimes you may have less not because of the resistance of the variety, but because of the level of toxin produced by the isolate.
Good. Ramjita, please.
Yeah, thank you, Farid, for this good presentation. The data you have shown for Mali, in Kolokani, the level of contamination is much higher than the other two sites. But I think you didn't go a bit further to elaborate on the reasons for that. The reasons, the factors. I would like to also refer back to the three projects you mentioned. Where do you think really we need to concentrate the work on those three groundnut projects in Mali, Nigeria, and Ghana in terms of aflatoxin?
Thank you. Yeah, I have a full report that I can give you a copy, because that we have been submitting a report to IFRI, and I can share the copy with you. And you can see in Kolokani, what we did here, we try to avoid to work with the farmers that we were working before, so that they don't know the aflatoxin management. Because otherwise, those people would apply whatever technologies we have been demonstrating. So that's why we really worked on the very far away villages, so that you have the real situation. And that real situation, Kolokani has got much more. But when you go to market in Kolokani, and the market near where we have been testing our technologies, and you can see very clear on that market the aflatoxin level is much lower. And we have regular sampling of that market. But you can see the data later. I think, USAID, the project that we were discussing with Rob last almost 8-9 months now, they want the FTF objectives to be achieved, and there is more yield, food security, and all those issues. So you have to, through this project, transfer those technologies. First step are going to assure food security. But at the same time, Rob was kind enough to accept that we have a component on aflatoxin management, part of those trials, and we did propose, and yesterday I sent the table. So there is a table of technologies that we can transfer in all these countries. In addition, the Nigeria project has got a component on aflatoxin management that, with the supplementary funding from USAID, that will give a boost to Nigeria.
Before we take up additional question, I think one announcement that scientific officers who need to leave for the transportation, they can leave, but all scientists, IRS and other staff, they can continue here. We may continue till 5 o'clock probably. So we can go directly from here to the happy hours.
Thank you.
Yeah. Okay, so now the next question is from Janila, and then we will come to you.
Thank you, Farid, for that wonderful journey. And I think with your vast experience, can you just tell us, although you have give us an overview telling about different research opportunities in this area, as an institute, what would be the top 2 research areas which you think in the next 5 years could really bring the difference to this area of work?
I think for an institute like ICRISAT, we should not have top 2. No, because it's very important. We have to have a vision long term. And that project, you can see there are short-term things that you can do, like the first package of technologies that you can give out and so on. And then you have a number of objectives, like what Kieran has been proposing, that would take anything between 5 and 10 years to go. They may be the best technologies to come. But as an institution, I think we have to have long-term things to keep going. Otherwise, we do short things, high priority, and then you lose the track.
I would like to know what is the impact of the groundnut cake being used for feeding to the livestock, where there is no monitoring of this aflatoxin at all. Are there any specific studies related on livestock?
Maybe Michael will reply to the livestock side, but what I can give you is the level of contamination or aflatoxin in the groundnut cakes is very high. It's almost concentrated because it's homogenized. In the case of seed, you can see some of them we just control, but in the case of cake, everything is mixed in. It's a very high level of aflatoxin. It's not only, in West Africa, for example, it's not just for animal, it's for human being. And we have been testing this in the case of Mali markets. People buy, and the Malians have got almost daily peanut sauce at home. And that peanut sauce sometimes could have very high level of aflatoxin. Michael, you want to?
I mean, it's correct. I remember that you did that study in another pool and found that more than 50% of the milk samples had non-permissible levels of aflatoxin.
Yeah.
A lot came from groundnut.
Good. Do you have some question from? I have a question from Shivali, and after that.
How to respond to this one? It's no, we did some work in the past. Honestly, that time we did not find. Well, there was a report that there is a resistance at the wild Iraqis, but to me, when I read the paper, I said, difficult to believe, because the seed of wild Iraqis is like stones. So how do you want the Aspergillus to go in? So once you bring it to the normal kind of eatable variety, then you can test it and find them. And we have been testing some of lines, but honestly, at this time, I have no idea of any high level of resistance.
Nice presentation. I don't know if it's working or not.
No.
Actually, I'm wondering, aflatoxin is a problem in some crops, but not a major problem in most of the crops. For example, in groundnut and maize, it's more a problem, and maybe to some extent in sorghum. But is there any specificity for this fungus, why it's more virulent or more damaging there, but not in other?
Oil, they like proteins.
But yeah, they are the protein crops.
Yeah. No, it's many of the oil seeds, you see, you take the case of pistachio, almonds, all these things are highly contaminated with aflatoxins. Yeah. Yes, exactly. Yeah.
I think what Farid didn't mention, that the aflatoxin work, just the way you talk about shuttle breeding, we are doing shuttle aflatoxin research between Malawi and here, and I think that is one of the things that needs to be noted, that the technology that was developed here, that we directly transferred to Malawi. We used to grind the samples and send here, and Farid will analyze, and they go back. But I think because we focus on breeding, we forget that there are other technologies that have been developed here that have been shared widely.
Yeah, thank you, Moses. There is a lot of things have been done, and honestly, difficult to report on every single part. But I think that what you did in Malawi, I think, is the best example of work that we should do more for with other regions. Yeah.
Yeah, thank you very much to my younger brother, for giving me. No, because we worked together for 35 years and always younger to me, he used to get excellent and clear on aflatoxin management. But when we go to farmers, quite often, say I'm the poor farmers, I take your advice, give some application and FOM and no stress management, moisture stress management, excellent. See, right now we don't have excellent varieties. See, like I take it with now, I think even 90.14 plus this package, good. But is there any quick way of detoxifying a spoiled situation? Can we rectify it? To almost like 10 ppb or 30 ppb to meet our export.
Yeah.
That's another thing. If we have something like whatever chemical treatment or dehydration and those things will be very useful.
No, I didn't touch on the detoxification. There are methods of detoxification for groundnut paste, but not for groundnut seed. Because ammoniation is the only technology that is possible to detoxify aflatoxin. So, where it is at the seed form, no. The other thing that people, the US guys have been developing is the clay technology. The clay technology is that you eat that particular clay, or you give it to animal feed. That has been approved by the US government, that they can add that small quantity of clay in the feed, and then that the clay absorb the toxin, and then it's rejected with this chemical. So they were thinking of putting this also in the human being food, but the government of US has not yet approved in the food system. I don't know in India. I think I'm sure that exists also in India. Many women, when they are pregnant, they would eat this kind of clay during the pregnancy. It's very common in my original country, Afghanistan. So those are part of things that exist, is the same kind of clay that has been there. Thank you.
Okay, thank you. Well, this is good. So anyways, I would suggest that we stay for a few more minutes. So
Yes. I would like to take 2 minutes to try to remember my peers. I think it's very important that we remember that we had a roots, and this pathology program, our groundnut improvement program, we had a roots, and there are a number of people. Sorry, I didn't want to do that. But I want to remember Dr. Duncan MacDonald, who passed away last year. He was the father of this lab and facilities we had, and he helped us moving and growing. So thank you to Duncan. And also, I don't want to forget all other Director Generals of ICRISAT. Dr. Swindle, Dr. Jim Bryan, Dr. Shauki Borkuti. Willie, I said that he has been giving so much of support. And many other colleagues here have been part of this effort. Duncan MacDonald, my brother Nigam was part of this all this time. Dr. Mahan, maybe I don't remember all the names right now, but I really would like to all of us to think of them. They have been part of us. They have developed many technologies for ICRISAT, and I really would like us to remember them and thank them forever. Thank you very much.
I am just sitting there. They will take over.
Anyway, I think as we heard that Farid has made very significant contribution to ICRISAT in the different areas, and as we know that one of his significant contribution is in diagnostic, aflatoxin diagnostic, which is considered or is one of our ICRISAT jewels, so this is really great. So of course, our Director General has already mentioned many times, but once again I would like to tell that research from Green Legume is very grateful to you. So please join me in thanking Farid Waliyar. And at this stage, I would like to invite our Director General, Dr. Dar, to Dr. Dar for here.
Yes. Come here. Hi, Farid, can you be requested to stand with us here? And before you receive the token of love, I would like to take this opportunity again to thank you for all the work that you have done, the small farmers of Asia and Sub-Saharan Africa, through ICRISAT. I have always mentioned all this time that our bosses are the smallholder farmers. They are the reason, the ether, why ICRISAT is here. And why the scientists are here, because you are needed by the smallholder farmers of Asia and Sub-Saharan Africa. Never forget that always. In everything that we do, you anchor yourselves to the very concerns and problems and of course the opportunities that you can give, uplift the human dignity of the smallholder farmers. Therefore today, how can we not bring them to become richer, get out of that trap of poverty? That will always remain a major responsibility of international organizations like ICRISAT. And so Farid, you have been part of that team. Team ICRISAT grows and will always focus its work, reminded of this need to transform subsistence farming, smallholder agriculture into prosperity. So we really believe that, yes, you have done so much for these smallholder farmers. I would have taken this opportunity to also remind Dr. Nigel Kirby from UK, who is a member of our board, to, they are the beneficiaries of the outcomes or the impact of the work of Farid in many ways in this aflatoxin kit. Again, to remind ourselves, in Malawi, for example, they stopped exporting peanuts because they have problems of aflatoxin. So 100,000 farmers, members of the National Association of Small Farmers of Malawi, stopped receiving more income from groundnut production because UK did not accept the aflatoxin levels that they have in their produce. But with now our intervention and partnership with NASPAM and Malawian government, they are back to the market. They're back to the supermarkets of UK. And so people of UK are directly benefiting from the work that we do in Malawi. And so they should be giving more contributions to the CG. They should be giving more grants to ICRISAT because they are healthier than anyone else. They are healthier, and so food safety becomes an increasing agenda in food and agriculture. So food safety research continues to give us that challenge, to see to it that we produce not just enough food, but very nutritious food, but also food that are safe, okay, and affordable to the poor and the smallholder farmers of Asian Sub-Saharan Africa. With this, Farid, thanks a lot for all this work. Can we give him a big, big hand?
Okay, can come in the center. Okay. But this is the center. Okay.
In my original culture in Afghanistan, when you have, they give you a gap, you have to put it in the shop. But in the French culture, they open it. Oh.
Aflatoxin kit.
Not an Eliza kit. This will go to my wife because she has a collection of the other things.
Okay, good. So thank you very much, Dr. Dar, and thank you very much, Farid. So now I think that I would like to invite you all for the happy hour, which I think we will be starting very soon in iMode court. Yeah. So thank you very much for coming to the seminar, and then to honor Farid.
The students.
Yeah.
Malawi.
Yes. So
Nigeria.
very good. Yeah. So yes. So once again, so thanks to all of you, and special thanks to our colleagues or students from Malawi, Nigeria, or from
Nigeria and Mali.
and Mali that they came here. So we really appreciate that. And thanks to all our colleagues in ES and WCA who have been participating in this meeting virtually. So thanks once again everyone, and have a great evening. And we look forward to see you in that happy hour.